Hello dear students, today we are going to study some important prime points from the chapter Microbes in Human Welfare. Okay, the first point regarding microbes in household products. You have to study only 3 points: production of curd from milk, fermentation of dough, production of cheese. First one, production of curd from milk. Lactic acid bacteria grow in milk and coagulate and partially digest milk proteins and convert into curd. The milk protein is called casein. This increases the nutritional quality as curd is enriched with vitamin B12. Vitamin B12 is cyanocobalamin. Okay, so the bacteria is called lactic acid bacteria, Lactobacillus bacilli. Then comes fermentation of dough. Saccharomyces cerevisiae, called baker's yeast, is used for fermenting the dough for bread production and for making foods such as dosa, idli, etc. So it is the yeast, baker's yeast or Saccharomyces cerevisiae. Then comes production of cheese. Propionibacterium shermani is used for cheese production, Swiss cheese production. And certain fungi are used in the ripening of Roquefort cheese. So 3 important products in household products. One is the curd, other one is the dough preparation, then comes the cheese production. Okay. The next prime point is regarding the industrial products. First one, production of fermented beverages. Beverages are produced by the fermentation of malted cereals and fruit juices using anaerobic microbes such as yeast in huge vessels called fermenters. The vessels inside which the fermentation occurs is called fermenter. Wine, beer, etc. are produced without distillation, whereas brandy, whiskey, etc. are produced by distillation. Here also the microbe used is Saccharomyces cerevisiae. That is why it is also called brewer's yeast. Then comes production of antibiotics. Antibiotics are chemicals produced by some microbes which can kill or retard the growth of other pathogenic microbes. For example, fungi, some special fungi are producing some chemicals which prevent the growth of pathogenic bacteria. Example, penicillin is a chemical produced from a fungus called Penicillium notatum. Another important industrial production with the help of microbes is the production of some chemicals and enzymes. The commercial and industrial production of acids, alcohols and enzymes mediated by microbial growth. For example, acid production. Citric acid is produced by Aspergillus niger. Acetic acid is produced by Acetobacter aceti. Lactic acid is produced by Lactobacillus bacilli. Butyric acid is produced by Clostridium butylicum. So all these are the microbes which are used for the industrial production of these acids. Then as you know, the Saccharomyces cerevisiae, the yeast, is used for the production of ethyl alcohol by fermentation. Then in the industrial use for detergent industry, some enzymes are used called lipases to remove the oil stains. And in the fruit juice industry, pectinases and proteases are produced to dissolve the fibers present in the fruit juices. Okay, all these are produced by the cultivation of microbes. Then another very important aspect is the production of some bioactive molecules from microbes. 3 important bioactive molecules are there. They are the commercial and industrial scale production of bioactive molecules. First one, streptokinase. It is produced by the bacterium called Streptococcus. It is used as a clot buster to remove the clots present inside the blood vessels. Cyclosporin A. It is produced by the fungus called Trichoderma polysporum. It is used as an immunosuppressive agent in organ transplant patients. Then comes statin. Statin is produced by the yeast or fungus called Monascus purpureus. It is used as a blood cholesterol lowering agent. So 3 major bioactive chemicals are produced by microbes. Streptokinase, acting as a clot buster, produced by the bacterium Streptococcus. Cyclosporin A, acting as an immunosuppressive agent, produced by the microbe Trichoderma polysporum. Statin, acting as a blood cholesterol lowering agent, produced by the microbe Monascus purpureus. Another important bioprime point in the microbes is the microbes used in sewage treatment. Sewage is the municipal wastewater produced in cities and towns which contain human excreta and other organic waste which are having abundant pathogenic microbes, highly polluting. The sewage is hence treated in order to reduce its polluting nature before it is discharged into natural water bodies. A polluted water means a high BOD. When we are treating the polluted water body, BOD means biochemical oxygen demand. The BOD will be decreased so that its pollution potential will be decreased. So two stages are there, the primary treatment and the secondary treatment. In the primary treatment is a physical process. The physical removal of large and small particles by filtration and sedimentation. After settling in the primary settling tank, the sediment form the primary sludge and the supernatant form the primary effluent. The primary sludge is totally discarded and the primary effluent is taken for secondary treatment. That means the sewage is, by primary treatment, get converted into primary sludge and primary effluent. Okay, the primary sludge is discarded. The primary effluent is taken for secondary treatment. Okay. Now the secondary treatment, which is a biological process. As you know, the primary effluent is taken for the secondary treatment. So here it is divisible into two sections, secondary sludge and secondary effluent. The secondary effluent is discharged into water bodies. Okay, the secondary sludge is used for biogas production. You can see the details. The primary effluent is passed on to large aeration tank, but is constantly agitated and aerated. This allows the growth of aerobic microbes into masses called flocs. That means the primary effluent is treated by microbes, aerobic microbes, so that the secondary effluent is produced. The primary effluent is with high BOD and the secondary effluent with low BOD. These aerobic microbes decompose the organic matter in the primary effluent, thus reducing its BOD. BOD means biochemical oxygen demand, which indicate the pollution potential, the capacity to pollute. After settling in the secondary settling tank, the sediment form the secondary sludge and the supernatant form the secondary effluent. The secondary effluent is totally discarded and the secondary sludge is taken as inoculum and also for sludge digestion by anaerobic microbes. So in the first step, for the conversion of primary effluent into secondary effluent, aerobic microbes are used. For the conversion of secondary sludge into biogas, or the decomposition of secondary sludge, anaerobic microbes are used in an instrument called anaerobic sludge digester. Okay, and the product is called biogas, which can be used for lighting and cooking. The next bioprime point is the production of biogas from microbes. Biogas is a mixture of gases containing methane, carbon dioxide, hydrogen sulfide, etc., produced by the microbial activity, which may be used as fuel. Certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with the carbon dioxide and hydrogen. These are commonly called methanogens, methanobacteria. These bacteria are abundantly seen in the rumen of cattle and also in their excreta called dung. Hence, dung can be used to generate biogas, which is commonly called gobar gas. So we have to study the structure of a biogas plant. A biogas plant consists of 5 components. The first one, digester. Digester is a concrete tank 10 to 15 feet deep in which bio-wastes are collected and a slurry of dung is fed. Then the digester is covered by a cover. There is a floating cover. It is placed over the slurry which keeps on rising as the biogas is produced in the tank by microbial activity. Then it has an inlet. An inlet tank is there for feeding cow dung and other bio-waste. And 2 outlets are there. Outlet number 1 for taking out biogas, it is a pipe. Then outlet 2, it is a tank for taking out spent slurry. So 5 components are there: the digester, a floating cover or lid, 1 inlet, 2 outlets. Okay. The next prime point is microbes can be used as biocontrol agents. Biocontrol refers to the use of microbes or use of biological methods for controlling plant diseases and pests. Biological control methods aim at the use of natural predators of pests and pathogens instead of using toxic chemicals as pesticides and thereby reducing the impact of atmospheric pollution. Example, ladybird beetles are used to control aphids, dragonflies are used to control mosquitoes. So here, microbes can also be used as biocontrol agents. Three examples are there. Bacteria used as a biocontrol agent, fungi biocontrol agent, virus biocontrol agent. First of all, bacteria. Bacillus thuringiensis is used to control butterfly caterpillars in brassica and fruit trees. Bacillus thuringiensis. Then trichoderma fungi, which are common in root ecosystem, can effectively prevent many of the plant pathogens. Then comes baculovirus of the genus Nucleopolyhedrovirus are pathogens that attack insect pests and other arthropod pests. So three types of biocontrol microbials, Bacillus thuringiensis bacteria, Trichoderma fungus and baculoviruses. Okay. Now come the last bioprime point in microbes. Microbes as biofertilizers. Biofertilizers are organisms, microbial organisms that enrich the nutrient quality of the soil. The usage of biofertilizers in agriculture is referred as organic farm. Then here also three types of microbes, bacteria, fungi and cyanobacteria. First of all, bacteria as biofertilizer. Some bacteria can fix atmospheric nitrogen as nitrogen compounds in the soil. This is called a nitrogen fixation, which can be readily usable by plants. Thus, they increase the nitrogen content of the soil. The nitrogen fixing bacteria can be both symbiotic and free living. The symbiotic bacteria include rhizobium in the roots of leguminous plants and frangia in the roots of non-leguminous plants. The free-living nitrogen fixers include Acetobacter, Azospirillum, etc. Okay. Then come fungi. Some fungi form symbiotic association with the roots of plants and they are called mycorrhiza. These fungal symbionts increase absorption efficiency, pathogen resistance, tolerance to salinity and drought of plants. Mycorrhizal formation seen among many members of the fungal genus Glomus. Okay. Then comes cyanobacteria. Cyanobacteria or blue-green algae are autotrophic microbes, many of which can fix atmospheric nitrogen. They add organic matter to the soil, thereby increasing the soil fertility. Example, Anabina, Nostoc, Oscillatoria, etc. Okay, so these are the biofertilizers, bacteria, fungi and cyanobacteria. So these are the main prime points in the chapter, microbes in human welfare. Okay, thank you.